there's a wealth of laboratory evidence that these anti-tumor properties kill cancer cells in a variety of ways. There are multiple mechanisms of action identified by which cannabis kills cancer cells, and they're divided into various categories. And among these are anti-proliferative effects. Normally, that's one of the hallmarks of a cancer cell, is that it just keeps reproducing. So if you stop the reproduction, that's an anti-proliferative effect. There are anti-angiogenesis effects, and this means that the cannabinoids will stop the tumor from being able to elaborate or grow new blood vessels to support the growth of the tumor. There are anti-metastatic effects, and that is simple enough to mean that the cannabinoids block the ability of the cancer cells to spread into other tissues. And there's another effect that has a wild name, apoptotic effect. Apoptosis refers to the ability of cannabinoids to speed the death of the abnormal cells. That's something that is especially important in cancer, because you're able to hasten the death of the cell without disturbing the normal cells around it.
Seth Research Laboratories in California have recently demonstrated that in some tumors, cancer cells are killed by marijuana, while the other healthy cells are left untouched. Cells that stop moving and become still white dots are dead cancer cells. The ability of cannabinoids to kill bad cells while protecting healthy ones is particularly important when we're talking about brain cancer because of the so-called blood-brain barrier. The brain has to be sheltered from outside influences that might hitch a ride on the bloodstream and cause havoc. What is exciting and unique about cannabinoids is that they can pass through the blood-brain barrier because of their slippery, fat-loving nature. Cannabinoids get right into the brain's cancer cells by moving easily through the cell's membranes, which are also composed of lipids. The evidence is piling up in mice-infested labs that the endocannabinoid system, when stimulated by cannabinoids, has an anti-tumor effect and can instruct cancer cells to commit suicide.
This was done by Manuel Guzman's group within the past less than 10 years, and what they showed there was that originally that THC, when injected into a brain tumor in mice and rats, a significant number of those animals the tumor would regress and disappear, so that you actually had survival of rats that would otherwise die. And they examined all the surrounding nerve tissue, and that was all fine, because remember, once again, cannabinoids protect nerves.
Dr. Manuel Guzman is a professor of biochemistry and molecular biology in Madrid, Spain, and is known for his groundbreaking studies on medical cannabis.
Hemos observado que los cannabinoides tienen un efecto inductor de la muerte de las células tumorales, un efecto inhibidor de lo que es el crecimiento, la multiplicación de las células tumorales. Lo que hacen es disminuir el crecimiento de los tumores.
If cannabis might be the miracle cancer cure that everybody's been searching for, then why don't doctors everywhere know about it?
People have a hard time believing that cannabis can have all of these fantastic effects that are described. But what we're doing is we're just stimulating a natural system that's already there. This has been developing for hundreds of millions of years. The invertebrates, the sea squirts, the hydra, there are primitive endocannabinoid systems in those organisms dating back 600-700 million years ago. The cannabis plant came along maybe 50 or 60 million years ago.
Why aren't billions in funds being directed toward cannabinoid research by the organizations that raise money for cancer therapies?
We're talking about medical research that, it's really a double-edged sword. On the one hand, I'm thrilled that there's really more research taking place now really than any time in history when it comes to the therapeutic use of cannabis, and specifically the cannabinoids, the components in cannabis. Unfortunately, a lot of that research is still relegated to taking place overseas. We see a little bit now finally taking place in this country, but really the United States remains fairly well behind the curve when it comes to cutting-edge medical cannabis research.
One of the first labs to demonstrate basically that not only the immune cells, the normal immune cells express these cannabinoid receptors called the CB2 receptors, but also that when these immune cells get transformed and they become cancer, to our surprise, we found that these cancer cells continue to express these CB2 receptors.
This was an exciting discovery because the CB2 receptor can act like a target for the cannabinoids. Once they bind with the receptor, they can tell the cancer cell to die.
So basically telling the cells basically to commit suicide, and that's what they do. And we demonstrated that that would be the mechanism by which the cannabinoids can kill the cancer, and therefore it can be used effectively as an anti-cancer agent.
Dr. Carte and his researchers were able to eradicate almost 100% of the cancer in test tubes. But they were skeptical they would see similar results when they moved on to tumors in mice.
To our surprise, we found that almost 25 to 30% of the mice completely rejected the tumor. They were completely cured. And in addition, we found that the remaining mice also, there was a significant reduction in the volume or the size of the tumors as well.
The lab results have been so promising that Dr. Carte is beginning clinical trials with leukemia patients. There's no doubt in his mind that the cannabinoids, either from the plant or lab created, will play a major role in medicine in the future.
I feel that in the next 5 or 10 years, there is going to be exponential growth in cannabinoid research.
It's an area where both the critics and the advocates agree. Scientists are now well on their way to developing medicines based on the cannabinoids. It begs the question, however, will modern medicine eventually make the marijuana plant obsolete?
No, it will not. In fact, it will only enhance it. Okay, because now it's more proof that the plant really does work. Okay, and instead of spending $600 a month on buying the pharmaceutical drug, I can grow my own plants.
I don't want them to ever take the choice away because I don't know how long it's going to be before they really find out exactly what is working for me or for others. And right now, having the raw plant available is the best solution because you have all of it there. You don't have just what they've isolated, just what they've decided is important now.
Let's keep it in the corridors of science. Let's keep it in the FDA. Let's do what needs to be done, which is careful, longitudinal, placebo-controlled, crossover, head-to-head studies, and see where it falls out. But let's deliver what's really medicine. That is the individual cannabinoids.
They may come across some things that are better than herbal marijuana for one thing or another, and good luck to them. I'd love that. But I never want to see compromised the capacity of people to use herbal marijuana, whether it's because the drug that they've come up with is much more expensive, or it doesn't do as well, or whatever the reason. People should always have herbal marijuana available to them without any constraints from the law.
Patients say for now the question is irrelevant. Science has not yet given them the opportunity or the plant to choose between an effective pill.
This is something where I have no other medical.
What is the evidence that marijuana smoking, habitual marijuana smoking, can lead to lung cancer? With respect to the development of lung cancer, we found no evidence of any increased risk of lung cancer occurrence in association with marijuana smoking alone. The marijuana smokers, if anything, had a reduced risk for developing lung cancer. Not a significantly reduced risk, but reduced less than one-fold, so that means reduced. Whereas the tobacco smokers had a markedly increased risk. If those who smoke more than 2 packs a day had a 20-fold increase in the risk. That's 2,000%. Those who smoke from 1 to 2 packs a day had an 8-fold risk. It's 800%. So that contrasts with no risk, no increased risk, or a slightly reduced risk with the marijuana smokers. THC actually has an anti-tumoral effect. And these are studies that were done both in experimental animals and in cell culture systems, and for different kinds of cancer. For lung cancer, breast cancer, thyroid cancer, prostate cancer, gliomas, brain cancer, that the development and growth, or the growth actually of the tumor is suppressed by THC, and metastases are also suppressed. So how can that be? Well, THC impairs protein synthesis, and it's what we call anti-mitogenic or anti-proliferative. You need, so tumor cells don't as readily proliferate in the presence of THC. They're also anti-angiogenic, so they interfere with the growth and development of new blood vessels that are necessary for metastatic spread. And they also are pro-apoptotic. What is apoptosis? Apoptosis is programmed cell death. So when cells age, there is a mechanism whereby the cells die. It's a non-necrotic death to die off the old cells, and we get rid of them before they have an opportunity to develop mutations that would lead to cancer. So enhancing apoptosis diminishes the risk of the cells becoming cancerous. So marijuana turns out, THC rather, turns out to be pro-apoptotic. So those appear to be the mechanisms that might account for these anti-tumoral effects of THC. We decided to do our own case-control study. Funding from the National Institute on Drug Abuse, which is a major funding agency for marijuana-related research. This was the largest study ever conducted on this subject. It was very well designed. We used the Los Angeles Tumor Registry to identify, rapidly ascertain all the cases of lung cancer and head and neck cancer that were diagnosed in the LA County system. And of course, by the time we got to some of them, they'd already died or were too sick, but we got to it over 60% of them who agreed to participate and were able to participate. And we administered this questionnaire, and then we matched them to controls, the same age, socioeconomic status, that lived in the same neighborhood, using an algorithm that USC developed for this purpose, so that we could match, we're comparing apples with apples. And then we administered this detailed questionnaire, the food frequency questions, occupational history, all kinds of things. We also did molecular, we got a buccal smear so we could look at the DNA, we look at the genetics of lung cancer. So what we did was to recruit smokers, heavy smokers of marijuana, at least a joint a day for a week, and it ended up that the average smoker of marijuana whom we recruited smoked 3 joints a day for about 15 years. And that's also required that they smoke that much for 5 years. But on the average, they smoked 3 joints for 15 years. So that's about 45 to 50 joint years. A joint year is the number of joints smoked times the number of years smoked. Over the study population was, I think, between 35 and 59. I think 35 was a younger age group. Of course, we thought that they had to be teenagers in their early 20s at the time of at least in the marijuana epidemic, which was in the mid-60s. So prior to that time, very few people used marijuana, but after that time, it just mushroomed up to 1979, which represented actually the apex, the acme of use of marijuana in our society. So that's why we chose those age limits. And so what did we find? For any category of cannabis use, including heavy use we define as more than 10 joint years, but we looked at 20 joint years and 30 joint years. For every category of marijuana use, the ratio was less than 1, meaning reduced risk. It wasn't significantly reduced, but it was reduced. And the confidence intervals were not that wide, around the point estimate. So there was no evidence, and we controlled for all the known or putative factors, for socioeconomic status, concomitant, tobacco smoking, alcohol, etc. At the same time, when we did a similar analysis for the tobacco smokers, there was a huge effect of tobacco.
We're now in the offices of Dr. Donald Taskin, noted pulmonary researcher here at the UCLA School of Medicine. Thank you very much for joining us today, Doctor.
It's my pleasure.
The show, of course, is Marijuana, Compassion, and Common Sense, and we'd like to talk to you about, you've done years of research with marijuana in relation to pulmonary systems. Is that correct?
That's right. Would you like me to tell you?
Yeah, tell us a little bit about
about my research and the results. I'm primarily interested in the health effects of marijuana, with the focus on potential adverse or harmful effects of marijuana rather than beneficial effects. Therefore, what I have to say may have some negativity to it in terms of the potential medicinal applications of marijuana use. But marijuana is smoked, generally. It could be eaten, but it's usually smoked. And it has very many of the same components in the smoke as are found in tobacco.
That was one of your research studies, wasn't it, to determine the carcinogens into marijuana smoke?
Well, our focus was actually to look not just at potential carcinogenic effects of marijuana, but also other possible health consequences, including chronic bronchitis, chronic obstructive pulmonary disease, impairment in the lungs, defense against infection such as pneumonia. So we did wide-ranging studies over the past more than 30 years. Now I could actually tell you in a nutshell.
I get that. First,
okay.
Why did you get involved in marijuana research? What interests you in marijuana research in particular?
What the results of those studies are, if you. Honestly, I was asked to look at the short-term, acute effects of marijuana on the lung by another investigator who had a grant from the National Institute of Mental Health back in the 1970s. And he felt, rightly, that since marijuana smoke is similar to tobacco smoke in many respects, and we know that tobacco smoke is harmful to the lung, that perhaps marijuana might share some of the same unhelpful consequences. So he asked me to look at short-term effects of marijuana smoking on the lung. We know that when you smoke tobacco, the airways, the bronchial tubes, tend to constrict immediately after smoking, and that constriction narrowing remains for several minutes, more than 15 to 30 minutes. So my first experiment was to see what happens to the size, the caliber of the airways after smoking marijuana compared to smoking tobacco. And I was amazed that after smoking marijuana, the airways actually opened up, dilated, in contrast to the effect of tobacco. So this piqued my interest, and we subsequently applied for some funding to look more carefully and in greater detail at the effects of marijuana on lung function, both the short-term effects and the long-term effects. That got me interested, and the more and more that we researched marijuana effects on the lung, we found important differences from tobacco that kept us going.
Well, now, what studies have you participated in that you feel kind of stand out as on your marijuana research? Because I know you must have done a whole bunch.
Right. We back in the 1980s, we recruited a large cohort of people from the Los Angeles area who smoked marijuana heavily. By heavily, we mean at least one joint a day for at least 5 years. And on the average, they smoked about 3 joints a day for 15 years. So it's pretty heavy smoking. We also recruited people who didn't smoke marijuana but smoked tobacco only. Smokers of both marijuana and tobacco. In fact, there were more of those than the marijuana-only smokers, as you might surmise. And also non-smokers of anything as controls. And there were about over 450 such individuals, and we have tracked them until the present time. So we performed a battery of studies on them initially, and we repeated some of those studies over time. Those studies consisted of just administering a health questionnaire to find out whether or not there was any impact of marijuana smoking on respiratory health. And we found that there was a much higher proportion of marijuana smokers, but marijuana only without tobacco, who complained of cough on most days for at least 3 months out of the year for 2 or more years. They wheezed on more than 21 days a year. They didn't have any increased shortness of breath compared to control. So immediately we could say from this study that marijuana smoking, heavy, regular marijuana, that's the definition of chronic bronchitis. They also brought up sputum. Now, and about a comparable proportion of tobacco smokers also had symptoms of chronic bronchitis. And we were surprised at that because the average number of tobacco cigarettes smoked per day by the tobacco smokers was about marijuana smoking is associated with chronic bronchitis. About a little over a pack, 22 a day, as compared to 3 marijuana joints a day. So there already was a 7-to-1 ratio, and yet there was comparability with respect to the impact on respiratory symptoms. We wondered about that, and that was the basis for some other work that we did. We thought that maybe the reason for the similarity in symptoms, despite the disparity in the amount of the plant substance smoke, might be due to a different way that the marijuana is smoked, might be due to more delivery of the irritants and gases and particulates in the smoke from marijuana than tobacco because of differences in the way the cigarette is prepared. We know that marijuana cigarette joints are loosely packed or not filtered, whereas tobacco cigarettes are filtered and more tightly packed. So, and what we found after tar is the insoluble particulate particle fraction of the smoke, which contains carcinogen. A series of experiments was about 4 times as much tar. These benzpyrene is the most potent of these carcinogens, and the 4 times as much was deposited in the lung. Compared from marijuana, single marijuana cigarette, compared to a tobacco cigarette of the same weight. Now, normally, marijuana cigarettes don't weigh as much as tobacco cigarettes. So that amplified the effect of tobacco, but that didn't appear to account entirely for this disparity. So I think that the question is not entirely answered, but maybe partially answered. We also looked at lung function. We know that if you smoke cigarettes, you have about a 20% chance of developing COPD, chronic obstructive bronchitis and emphysema, which when it is severe is associated with symptoms of difficulty breathing on exertion and results in an increased mortality, not only from COPD itself, but also from heart disease. So we performed the test of lung function. We were surprised. We found that there was no abnormality that we could detect in the lung function of smokers, heavy smokers of marijuana, but there were abnormalities in lung function in the smokers of tobacco only, and there was no additive effect. So we thought, well, maybe there would be a change over time, that we know that COPD is characterized by an accelerated rate of loss of lung function over time. It's a progressive disease. Well, maybe we'll see that if we repeat the lung function tests year after year. We did that in the marijuana smokers and the tobacco smokers and the controls. And lo and behold, we found that whereas lung function continued to decline at an accelerated rate in the tobacco smokers, it did not in the marijuana smokers. And that led us to believe that for one reason or another, marijuana smoking, even heavy smoking, does not appear to be associated with the development of COPD. It may not be a risk factor for COPD. Now, there are some other data in the literature that contradict that view, but those were our findings. That interested us. I have some theories as to why that is the case, but we might.
Okay. Yeah, I would love to get to that, but so the other study, of course, the reason I hear is because of the recent study on the connection between marijuana, tobacco, and lung cancer. And I was wondering, how did you come to do that study?
Okay. Yes, get to that later on. That's a very good question. We know that tobacco smoking is the most important cause of lung cancer and also head and neck cancer. Marijuana smoke contains much the same ingredients as are found in tobacco smoke, including a number of carcinogens, vinyl chlorides, polycyclic aromatic hydrocarbons like benzpyrene, phenols, nitrosamines. It contains billions of oxyradicals that cause oxidative stress that predisposes to carcinogenesis. So it's not an unreasonable hypothesis that if you smoke enough marijuana, that you may be at increased risk for developing respiratory cancer. That's number one. Number two, I already mentioned that the exposure of the lung to the carcinogens, the tar, which are contained largely in the tar fraction, is magnified by the fact that about 4 times as much of the tar is deposited in the lung from a marijuana cigarette compared to a tobacco cigarette of the same mass. That's number two. Number three, there are studies in cell cultures, tissue explants that show that exposure to marijuana, or lung tissue explants, that in the test tube, if you will, results in accelerated malignant change. Mm-hmm. We know that marijuana smoke is mutagenic, and that mutations lead to cancer development. Then there are studies that we carried out extensively in marijuana smokers and controlled non-smokers, as well as in tobacco smokers, in which we inserted a lighted scope called a bronchoscope into the airways of our subjects, and we obtained biopsies of the tissue lining the airways, and we were surprised to find widespread changes, abnormalities, microscopic abnormalities in the cells that lining the airway, some of which are known to be precancerous. And the extent of abnormality was similar in the marijuana smokers compared to tobacco smokers, despite the disparity in the amount smoked. Mm-hmm. And then I won't bore you with the names. There's squamous metaplasia. We also looked at abnormalities in the nuclei. We also looked at molecular markers that would predict development of tumors and even more aggressive tumors. Ki-67, it's a nuclear proliferation factor, and epidermal growth factor receptor are two of those that we looked at. They were markedly overexpressed in the airways of marijuana smokers compared to non-smokers, even more than in the tobacco smokers. So that led us to believe that it's plausible to expect, to hypothesize, that there might be a link between marijuana smoking and cancer. Also, there are studies in man. There are case series of young people with either head and neck cancer or lung cancer in whom marijuana smoking is overrepresented. In some of these series, up to 90% of these individuals, it's rare to develop cancer when you're young, but over 90% in some series were marijuana smokers, a much higher proportion than in the general population. Also, there are some epidemiologic studies, even one of ours, that showed an increased risk. These are case control studies for head and neck cancer. On the other hand, there are other epidemiologic studies that show the opposite, or fail to show an association. Finally, we developed an animal model of lung cancer. It's a mouse model. And we found that if we implant lung cancer cells into the flanks of these animals, and then we treat some with THC, the major ingredient, psychoactive ingredient marijuana, and some with a vehicle placebo, if you will, those who were treated with THC developed much more aggressive tumors that grew at an accelerated rate. So all this evidence led us to believe that there would be an association. But the bottom line is, a mouse is not a man, and is that you can't say that there's an association unless you demonstrate it in people who smoke the way they normally smoke. And that's the reason we carried out this study.
And could you tell us
the results?
The results of the study?
Yeah, the study was carried out. It was a case control study. The principal investigator was Dr. Hal Morgenstern, who's a professor of public health. He used to be here. He's now a chair of epidemiology at the University of Michigan. And he's an expert, a world's expert in case control methodologies. It was very carefully conducted. We ascertained all cases diagnosed with lung cancer and head and neck cancer in Los Angeles County over a few year period. Rapidly ascertained these diagnoses and then attempted to contact all of these individuals. We succeeded in a little over 50%. Some had died before we were able to contact them. Some were too sick to participate, etc. And then we actually also contacted controls who were matched on age, gender, and socioeconomic status, and the residents, the neighborhood. They had to be in the same neighborhood in close proximity to the case. So we actually succeeded in studying about 600 lung cancer cases, about 600 head and neck cancer cases, and over 1,000 matched controls. We administer to every cases and controls the same extensive questionnaire that gets at their history of smoking, anything, tobacco, marijuana, crack cocaine, etc., use of other drugs, other known or putative risk factors for cancer, such as a family history of cancer, diet, occupational exposures, etc., etc. And so from that information, using rather traditional statistical approaches, you can determine what the relative risk is, or the odds for developing cancer, either lung cancer or head and neck cancer, in relation to, in association with marijuana, controlling for all the other factors, tobacco, alcohol, socioeconomic status, age, etc. And the bottom line is, we failed to find any positive association between marijuana, even heavy marijuana use, which we defined as more than 10 joint years. Even if you look at more than 60 joint years, a joint a joint a day, times the number of joints smoked, we could not see an association, nor did we find any potentiation of the carcinogenic effect of tobacco, nor did we find any reduction in the risk from tobacco. So there was no interaction with tobacco. If anything, the risks were a little bit less than one, but not significantly so. And I don't think there really is protection, because if there were, one would see a dose-response relationship. That is, the heavier smokers would show more protection than the lighter smokers, or lower odds ratio, if you will, and they didn't show that. So I think it's really essentially a negative study. We failed to find any positive association with cancer. Now, what does that mean? Does that really mean there absolutely is no association? We don't know that. I mean, it is possible that people with the appropriate genetic makeup who are particularly susceptible to lung cancer, if they're exposed to any carcinogens, whether they be in marijuana smoke or in some other substance they're exposed to, might be a greater risk. We can't exclude that possibility, but our data are unable to answer that question.
Well, now, the so-called protective effect, the group that there was some, actually the people who smoked marijuana had a somewhat less development of lung cancer than the people who didn't.
On the average, but not significant.
Yes,
so
but,
not statistically significant.
But yet, the people who are smoking marijuana are being exposed to these carcinogens, and for some reason, they're not developing the cancers that they should be, because, like tobacco smokers do, who are exposed to the same thing. So there's some. I know Dr. Abrams, I know, asked you at the conference, is there a protective effect? And you responded that was not an unreasonable hypothesis. What did you mean by that?
It's still an hypothesis. Yeah. We know that there are, in some studies, anti-tumoral effects of THC. Not in all. In other studies show a pro-tumoral effect. Right. And these are studies that were carried out in a variety of animal models and cell cultures. And what they show is that when you expose these animals or cells to THC, there is a reduction in tumor growth, the rate of tumor growth, and also a reduction in spread, metastases. And three mechanisms have been proposed. One is an anti-proliferative effect of THC, impairment, let's say, in protein synthesis, etc. One is a so-called anti-angiogenic effect. In order for a tumor to grow and spread, it has to develop a blood supply. And it appears that THC inhibits the development of that blood supply. And another is something called a pro-apoptotic effect. That means an enhancement of early programmed cell deaths. So that cells that age ought to die off early in order to prevent them from undergoing malignant change as they age. So some studies have shown a pro-apoptotic effect. Other studies have shown an anti-apoptotic effect. There are other animal studies that show an anti-tumoral effect. So you could hypothesize that perhaps what's really operative in these cases is an anti-tumoral effect that counterbalances the pro-carcinogenic effect of the carcinogens. Mm-hmm. So that's a possibility. That doesn't mean if you smoke you're going to be less likely to develop it. It means if you smoke, you may be counteracting the cancer promoting effects of the reasonable hypothesis.
Well, Doctor, I've got a lot more questions to ask you, but today's interview we've run out, so we're going to close this one down and we'll bring you on for our next show next week.
Okay, real fine. Of the smoke. That's purely hypothetical. That's why I said it's not